Hey students, welcome to my channel, The Coach. I am back with another question which has been asked in NEET 2023. So the question is, which hormone promotes internode or petiole elongation in deep water rice? So the options are first option carnitine, second ethylene, third 2,4-D, and fourth is gibberellin, GA3. Let's start discussing the option. First option is carnitine. So carnitine is a plant hormone. Carnitine is a cytokinin which has a greater role in cytokinesis. Cytokinesis means what? Cell division. Cell division. Cytokinesis is division of cytoplasm. So during the cell division, the first step is when nucleus divides, and after the nucleus division, cytokinesis takes place where the cytoplasm divides, right? So the cytokinin helps in the cell division, promotes cell division and helps in cytokinesis. Next role is overcome the apical dominance. So you can see here, apical dominance is the phenomena where the active apical bud keeps on growing, and due to the fast growth of the apical bud, the auxiliary buds, or you can say lateral buds, the lateral buds stop its growth, due to which plant grows in length but it does not become very bushy. So cytokinin helps to overcome this apical dominance and leads to the growth of lateral buds, which makes plant dense and bushy. Now, next role is promote nutrient mobilization, which helps in delay of leaf senescence. So leaf senescence is, you can see this is senescence leaf, right? Death of leaf, aging of leaves. Cytokinins delay the senescence of leaf. Now let's look at the second option. Second option was ethylene. So ethylene promotes rapid internode or petiole elongation in deep water rice plants. So what happens? Rice plants, you can see here, is a plant which is mostly grown in water. Okay, so most of the part of the rice plant in the initial stages is submerged in water. So what happens is when you put ethylene or under the influence of ethylene, ethylene again is a plant hormone, the petiole or the stalk, or you can say the internode of the rice plants gets elongated. So what happens? Rice is a monocot plant. So in the monocot plant, you can see here there is no petiole. The leaf base, this is the leaf base which extends to form a sheath like structure. So here, how the length of the monocot stem increases? The length of the stem increases by expansion of nodes and internodes. So expansion, so the length of the monocot stem increases by expansion or elongation of internode. So here is one node. Let's say here is one node. So when this area elongates in the presence of ethylene, the length of the monocot plant increases. So in the rice plants, what happened in the initial stages, when the rice plant is very small, it is submerged in water. Okay, so if this is the rice plant, it is submerged in water. Once the petiole elongates, it comes above the water, and due to which it is able to do the gaseous exchange. So this is the main purpose of elongation of internode. So ethylene not only does this, but ethylene also promotes the rapid growth in the plant, which increases the absorption surface of the plants, monocot plants or any plants which is under the influence of ethylene. Now let's look at the third option, that is 2,4-D. 2,4-D is a auxin. It is basically a synthetic auxin. So this auxin is basically used to make weed-free lawns. You can see here this side, weed-free. There is no weed. Weeds are unwanted plant which grow in between the main crop. So here the main plantation is of grass. In between these weeds occur and they grow and they compete with the plant for nutrients, due to which the growth of main plants decreases. So 2,4-D is a synthetic auxin which is used to kill these weeds. But these weeds only if they are dicots. Only if they are dicots, then it gets affected by 2,4-D. But if it is not a dicot, if it is a monocot, monocots metabolizes the 2,4-deoxane, because 2,4-D monocots metabolizes herbicides like 2,4-deoxane and remains unaffected. So if the dicot weeds are removed by 2,4-deoxane, then let's look at the last option, that is gibberellic acid, GA3. So GA3 is basically used to speed up the malting process in brewing industry. So brewing industry where beer is made by using cereal or grains. Okay, so in the brewing industry, there is a process called malting. Why malting is done is that during the malting process, before making converting the cereals or grains into beer, we have to do malting. Okay, so malting is the process by which the grains get aware. You can say by the process of malting, the enzymes which is present in the grains which is used to prepare beer, that enzymes gets activated, and due to which the complex carbohydrate which exist in malt, like maltose, like complex carbohydrate, for example maltose is broken down due to the activation of enzyme in the malting treatment. Maltose is broken down into glucose and glucose. So when in during the production of beer, the microbes or fermentation takes place, that time the microbes which ferments the beer gets a good amount of carbohydrate which is used up. Okay, so malting is a process. See here on the left side I have prepared given a diagram. This is steeping, where you put the grains in water, then you germinate the grains. By the process of germination, due to the germination, you allow the conditions so that grains get start sprouting or germination, so that all the enzymes gets activated on germination, and finally you do kilning process where the roasted rice gets specific color and taste. Then now this after malting, the grains go for production of beer in industry. So if you use gibberellic acid, the malting process gets speed up, and it enhances the content of simple carbohydrates into the grains, due to which it is very easy, the carbohydrates are easily available for the microbes for fermentation. So from the above discussion, we can conclude that the correct answer is ethylene, because ethylene helps in internode or petiole elongation. Thank you.